afternoon to all. Today we are going to discuss a new topic, that is protein synthesis. That means to create proteins by using the genetic information which is encoded in the DNA. Mainly the protein biosynthesis have two stages, that is transcription and translation. Transcription means the mRNA which is produced from the DNA molecule, that is deoxyribonucleic acid. It is as a genetic material, whereas translation is a process by which proteins are produced from the mRNA molecule. Both the transcription and translation are three steps, that is initiation, elongation and termination. Then the process are divided into two, that is this one is the transcription and this process is the translation. So mainly an enzyme play an important role in the process of protein synthesis, that is RNA polymerase. The enzyme which is RNA polymerase. It is an enzyme which is used to synthesize RNA. So I told you before, this is the entire process which is happening in the transcription process, that is initiation, elongation and termination. So in initiation process, we know that this is the DNA molecule, that is genetic material, deoxyribonucleic acid is there, and this DNA which is inside the nucleus. This is the nucleus portion and which is inside the nucleus, and inside the DNA there is a particular region, that or the region. Mainly the promoter region can help the process of transcription, and the RNA polymerase, which is an enzyme, that enzyme binds to the promoter region of the DNA molecule that help to produce the RNA strand or mRNA molecule. Then in the case of elongation process, I told you before, the RNA polymerase is bind with the promoter region, it produces the new complementary DNA strand. So the new complementary DNA strand is there. At the process of termination, then the specific DNA sequence that detach the process of termination, and the RNA polymerase enzyme will be detach the DNA. So this is the overall structure about the transcription process. And I told you before, the transcription process happens in the nucleus, but in the translation process it will be happen out of the nucleus. That means the mRNA transcript go to the cytoplasm, and inside the cytoplasm contain the cell organelle which is ribosome. So ribosome play an important role in the protein synthesis. I told you before, there are three stages which is involved in the process of translation, that is initiation, elongation and termination. So ribosome can control and coordinate the three steps which is involved in the translation process. Okay, so in the termination process, I told you before, it happens in the ribosome. So the mRNA molecule which is bind to the ribosome and it produces the amino acid sequence. That means in the mRNA molecule they have a small molecule which is known as tRNA molecule. That tRNA molecule can help to produce the codons of amino acids and which bind to the mRNA molecule. Then the ribosomes moves along with the mRNA and it leads to the codons, and finally it leads to the amino acids in a polypeptide chain. This is actually happens in the elongation process, and when we go to termination process, the process will be stopped when the ribosome encounters the stop codons. That stop codons are UAA, UGA and UAG. These are the stop codons. These stop codons can help to the translating the mRNA and it helped to release the protein.